Hello, everyone, and welcome to today's Life Science and Translational Research presentations. I am Xavier Gutierrez of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is sponsored by Agilent. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Send. Please address each of your questions to a specific speaker. We will answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Ask a Question box located on the far left of your screen. I now present today's speakers. First, we will have Dr. Stephen Pennington, founder, CEO, and CSO of Atturos Ltd, present on the topic of personal protein signatures for precision medicine. Then, Dr. Clifford Dacso, Professor of Molecular and Cell Biology at Imperial College of Medicine, along with his co-presenter, Dr. Arun Sreekumar, Professor and Director of Metabolomics, Department of Molecular and Cell Biology at Imperial College of Medicine, will present on rewired metabolism in diseases, biomarker and therapeutic applications. For a complete biography on today's speakers, please visit the Biography tab at the top of your screen. Dr. Steve Pennington, you may now begin your presentation.
Good day, everybody. I'd like to start by acknowledging my thanks to Agilent for giving me this great opportunity to present at ASMS. And I renamed the title of my talk Testing Times because, of course, we are in very testing times indeed. And I think nobody would have anticipated at the beginning of the year that we would be having an ASMS that was virtual and that we wouldn't get the opportunity to meet in person. But I've heard that ASMS is going extremely well, and HUPO likewise is going to have a virtual conference and congress later this year. And we just hope it can be as successful as ASMS and this event as part of ASMS. Of course, when I put these slides together in May of this year, nobody realized that having been hit with a pandemic, a viral pandemic, the world then was also going to be exposed to a reemergence of concerns over racial inequality. So these really are indeed testing times, and I appreciate that all of us are going through what is a quite remarkable experience. So I hope that you are all safe and well. What I'd like to do in my presentation today is talk about the development and delivery of advanced protein tests for precision medicine. And much of that work has taken place in the Conway Institute, which is the building that you can see in the slide. And at the bottom of the slide, you can see some of the funding agencies that have supported the research that we've been doing. I think it's really important to acknowledge at the outset that part of the work I will describe and the continued development and delivery of the tests that we're trying to produce is being done in the context of a small spin-out company, UCD-based spin-out company, that's University College Dublin, and that company is called Atturos. And Atturos really wants to develop protein data for precision medicine to deliver advanced diagnostic tests and the data from them that will have a global impact. Of course, COVID has affected all of our lives, and I suppose what I've tried to do here is just capture some of the emerging lessons, because one of the things I tried to do early on during the COVID crisis was really to try to look for some of the positives, if there are many or any. And I suppose one of the things that we've all got used to dealing with is a new degree of uncertainty and flexibility. And of course, we've all been really conscious of the impact of external factors, how plans and research plans can be really changed dramatically by factors that we hadn't been able to anticipate. I think one of the positives, and certainly from a public perception, is the importance that it's raised on testing, both DNA and protein tests. And of course, being able to do testing at scale. So I think science has benefited from a public realization that testing is really important. But of course, we're also conscious that these tests need to be reproducible and reliable. And I suppose there's a possibility that some of the tests that are being developed rapidly may not be as reproduced, COVID tests may not be as reproducible or reliable as we would like them to be. I think the other thing that I particularly realized is that, of course, once there's a will to do something, there's enormous energy can coalesce around that will, And as my granny used to say, where there's a will, there's a way. And it seems that we have moved forward quite remarkably in the last few months. But we've moved forward in the sense of being able to test for COVID and possibly being able to manage and ultimately treat this viral disease. But of course, chronic diseases, the existing diseases that individuals have, haven't gone away. And as we come out of the COVID lockdown in various jurisdictions, it's becoming obvious that the chronic diseases and patients with chronic diseases have been slightly neglected for the last few months. And that's something that we will have to deal with, amongst many other things. So what I'm going to try to do in the brief time that I got available is give a sort of an introduction to precision medicine, a very brief, talk about the early development of protein biomarkers, and then how we might deliver those protein biomarkers specifically for some chronic diseases. And I'll end with some observations. So this gentleman, Leroy Hood, has a vision for precision medicine, and this is him articulating that vision at the 2017 HUPO Congress that was held in Dublin to an audience of about 1,500 delegates. And in Leroy Hood's vision of medicine, P4 medicine as he calls it, the future of medicine will be predictive, more preventative, it'll be very personalized and participatory. And so he envisages that systems medicine driven by the omics and the large data that's generated as a consequence, driven through a digital revolution and social networks, will lead to this new era of medicine. What's interesting from my perspective is that Leroy recognizes that many new platforms will have the potential to improve health of individuals. In particular, platforms for proteomics will be important. And Leroy suggests that's the case because proteins are the active agents in cells, and so measuring proteins gives us a much closer handle on physiology, and I think many of us appreciate and understand that. But he also acknowledges that realizing the potential of P4 medicine presents a number of challenges and at very many levels. So that's the vision for precision medicine of the future. What about medicine now? Well, we know that it's very imprecise. And this figure just shows, and I'm sure many of you will have seen it before, where there's a patient in blue, that's a patient who's been given a drug and responded, and in red, a different patient will have been given the drug and not responded. And this is a list of the top best-selling drugs in the US. And you can see that the very best is effective in 1 in 4 patients. So that's medicine as it stands now. And so in many cases, these top-selling drugs are only effective in 1 in 15 or 1 in 16 patients. And of course, the 4 or 15 or 16 or however many it is patients who don't respond, not only do they have the side effects of the disease, but their disease is progressing. So that's a double negative hit. And it's been suggested by Yamamoto and colleagues that whilst genomics is extremely exciting, and we were all excited about the sequencing of the human genome and the genomics revolution that's occurred subsequently, genomics in itself, no matter how powerful it is or economical, isn't sufficient to understand human physiology and pathophysiology. And they suggested that we would have to have this knowledge network of data from different sources: genetics, the microbiome, exposures, patient-contributed data, as well as new clinical tests to be able to identify patients who will respond to particular treatments or given particular therapeutic paths to follow. And it's our hypothesis that the clinical tests that they propose could be new protein biomarker tests, and that these protein biomarker tests will be complementary to genomics and add value to it. And so obviously these new tests are going to have to reflect the heterogeneity of individual diseases and the variability of individual patients, including their co- and multimorbidities. So many individuals with chronic diseases are multimorbid. And of course, we all know that environmental exposure and lifestyle behaviors influence health. And so the clinical tests at some stage are going to have to incorporate these important factors into their reckoning. So combining proteins data and genomic data for some is the emerging field of proteogenomics. And here we have a picture of Joe Biden talking on proteogenomics and his conviction that proteogenomics will accelerate the pace of cancer research. He did that in 2017 at the Royal College of Physicians in Dublin. Joe Biden's view of the world is that we, as a scientific and clinical and medical community, need to speak clearly and engage with public and patients. we need to change the way that we do things, change our culture. But most importantly, he implored during this talk and has done subsequently that we should have impact, that is, we should do something to benefit patients, and that we should do it with a sense of urgency. So he firmly believes that you, that's us, have a huge opportunity with the emergence of this new omics era and responsibility to do something, to use that data to patient benefit. So, of course, what we would like to do is to improve patient outcomes using proteomics. That is, in Biden's words, have some impact. And I suppose one of the ways we believe that we might be able to do that is by identifying better biomarkers, and those better biomarkers would be converted to better tests. Of course, we're all patients, and I'm sure many of us will have had this experience, a venous blood draw, where blood is taken and processed and then used to support the performance of a diagnostic test. So this is a test form from a hospital in Dublin, St Vincent's Hospital. And on the right-hand side, there are some tick boxes for tests that are going to be performed. So venous blood draw is taken on the bottom left-hand side, the sample processed and then taken to a laboratory where the test is performed. And on this test, there are on this form, there are tests for individual proteins. And one of these proteins is PSA. Many of you will have heard of it, prostate-specific antigen. So this is a single protein test that's measured in blood. And indeed, many of the diagnostic tests on this form are for single proteins measured in blood. In the case of PSA, that protein is measured on an automated ELISA as part of a diagnostics clinical track, and so in a very automated process. However, PSA, although it's used for prostate cancer diagnosis, and there are over 30 million PSA tests per year performed in the US alone, a high PSA doesn't mean that an individual man has prostate cancer, and a low PSA doesn't mean that he doesn't have prostate cancer. So for diagnosis, this single protein test, although it's performed so widely, isn't good, and most people know that. And it's also for prognosis, that is for predicting the outcome of the cancer in the patient, it's actually worse. And I think the similar limitations apply to all of the single protein biomarker tests that are currently used by clinicians to identify disease status in individuals or diagnose diseases in individuals. So what I'd like to do now is just talk about how we might improve this situation by the early development of protein biomarkers. And many will have seen a schematic like this before, where we begin with protein biomarker discovery. We identify some candidate biomarkers, develop an assay to confirm that those biomarkers do really exist and change under the conditions that we're measuring, and now assemble them in a panel, not as individual biomarkers, but as a panel, and perform an evaluation of the performance of that panel in the hope that we might ultimately get to a new clinical test. That can be split into phases of discovery, developing the assays, using those assays to evaluate the performance of the biomarkers, and then moving them to some phase of clinical utility. I just want to emphasize that the analytical validation step is extremely important, because what we need is a reliable and robust method of measuring the proteins. And if we don't have that, then ultimately we're not going to have a test. There won't be any clinical utility. So what's the strategy for doing that? Well, within Atturos, we held some brainstorming sessions on a range of topics of clinical, technical, and business development aspects of trying to produce new biomarker tests. And of course, the three key elements are the content of the test, the platform, and its intended use. But then there are a whole series of questions. And I suppose this alludes to the complexity that Leroy Hood was talking about and the challenges. And we know it's challenging. There have been reviews on how futile efforts have been. Some of the known challenges have been reviewed in numerous manuscripts. So we know that it's a very challenging process. So I hope you can imagine my surprise a few years ago when a young bright-eyed consultant from Cambridge, Dean Griffiths, said, actually, if you want to develop and commercialize biomarkers, it's really simple. All you've got to do is identify a need, come up with a solution, and scale it. And of course, he was right. And so in our case, that would be identify a need, discover and develop the biomarkers, and then deliver them for their use. And that made us realize that what we really needed to do if we were going to try to develop new tests was to start at the end. Unfortunately, I don't think this slide has projected. There should be a very large start at the end box in the middle of the slide. And by that, I think, oh, sorry, there it is now. By that, I think what we need and mean is that we have to start with the clinical unmet needs and identify that need and the clinical test that will address the need, and then develop and evaluate the biomarkers against their intended use. And of course, as basic scientists, we're not in best place to understand what the clinical needs are. And I think that requires a very close association with clinicians and who work with the patient groups that we're interested in or in chronic diseases. So I'm just going to change tack slightly, and I just want to talk now about what I think has transformed or has the potential to transform our ability to develop new biomarker tests. And that's peptide multiple reaction monitoring. I'm sure many, if not all of you, know exactly what peptide MRM is. That's identifying a peptide as a surrogate of a protein and quantifying it in a triple quad mass spectrometer, predominantly a triple quad mass spectrometer. And we think, as do many others, that MRM will play a key role in developing analytically validated measurements for protein biomarker candidates, and then enabling the clinical validation of those multiple protein biomarker candidates. What happens then, and has happened in a number of studies, is that once the clinical validation has been performed and a small panel of protein biomarkers has been identified, individuals have then developed ELISAs to use those ELISAs as a clinical test and so support clinical utility. And in many ways, that seems a really sensible thing to do. But I think for reasons we can't spend time on today, I think that it actually isn't. And the reality is that it's proving very difficult to develop ELISAs, multiplexed ELISAs, to proteins that have been discovered and validated and clinical validation undertaken using mass spectrometry-based methods. So I suppose ultimately our hypothesis is that MRM will be the clinical test platform, as well as being the platform on which the protein biomarkers are evaluated and clinically validated. It will be the platform under which the tests are performed. Of course, MRM is very popular. We developed our first multiplexed MRM assay in 2006, and so we've been doing it for a while. And I think there's still, from my perspective, a little bit of, not sure how to put this, a bit of over-expectation about what an MRM assay is. So it is undoubtedly an ability to measure a peptide, that peptide's measured in a sample that mass spectrometrists may refer to as a matrix. It requires liquid chromatography to separate peptides and mass spectrometry to detect and quantify them. And an MRM assay, if it's going to be effective, needs a lot of quality control. And I think one of the things I'd like to try to emphasize is that what an MRM assay, once you've identified a prototypic peptide and transitions that you may want to measure in the mass spectrometer and a retention time under various liquid chromatography conditions, it isn't a universal assay for the quantification of that peptide or protein. It's very, very dependent on the sample matrix and any pre-analytical steps that you may have taken with the sample. So we spent a number of years and now transferred this to Atturos, developing a protein biomarker platform. And the biomarker tests that we're trying to develop are we measure multiple blood proteins, do it simultaneously and precisely. The workflow supports accurate and reproducible data, that is, that they are analytically validated. The data that's generated is processed by machine learning approaches, predominantly random forest approaches, and we then develop an algorithm that allows us to create an easy-to-interpret score for whatever distinction we're trying to establish. And within Atturos, that now is a protein biomarker workflow, and we're enormously grateful to Agilent for them enabling us to develop this workflow, in which everything is carefully controlled. The sample tracking and processing and pre-analytical steps are extremely important. That the processing then is done in an automated way. We're talking here about tryptic digestion, and then those samples go on to triple quad mass spectrometers, predominantly the 6495 series of mass spectrometers, and the data analyzed by machine learning approaches. And where we can, the data is then integrated with other clinical data. And all of this requires a series of mass spec process controls and pre-analytical method controls. So a lot of standardization. So this is really, at this stage, it's more product development than it is research. So what I'd like to do now in the brief time that I've got available is just talk about delivery of some protein biomarkers for chronic diseases. And I'm going to focus on three areas that Atturos is working on. That's prostate cancer in the middle, psoriatic arthritis on the left, and kidney disease on the right-hand side. In the case of psoriatic arthritis, the current average time to diagnosis of that disease is 8 years for a precise diagnosis. That means that patients aren't treated appropriately for a very long time after their initial presentation to a rheumatologist. In prostate cancer, we estimate that at least 300,000 men per annum globally could be spared invasive treatment for their disease. That is, they could go on something that's called active surveillance. And in the case of diabetic kidney disease, in the case of diabetes, about 30% of patients go on to acute kidney failure. So these are identified and significant clinical unmet needs. And what I'd just like to do now is just pick one of those, and that's psoriatic arthritis, and just mention in a few slides what we've been doing in the domain of psoriatic arthritis. So we're very fortunate. I think this slide has been slightly scrunched, but in the domain of psoriatic arthritis, we've been very fortunate to work with a clinician, Oliver Fitzgerald, who's one of the world-leading clinicians in psoriatic arthritis. He's the incoming president of a group called GRAPPA, an unfortunate acronym, but it stands for the Group for Research and Assessment of Psoriasis and Psoriatic Arthritis. It's around 1,000 dermatologists and rheumatologists worldwide, and Oliver, as I say, is the incoming president. Psoriatic arthritis is a complex and polygenic autoimmune disease, and it has diverse clinical features. And you can see some of those features here, ranging from uveitis, inflammation of the eyes, spinal involvement. It is associated with psoriasis, so about 30% of patients with psoriasis will go on to get psoriatic arthritis. It's also associated with pitting of the nails, dactylitis, that's swelling of the digits, and very characteristic joint erosions. In its most severe form, psoriatic arthritis mutilans, you can see that the deformities that it induces are life-changing. Individuals with these kinds of bone and joint changes really can't operate and function in a normal existence. Most patients with psoriatic arthritis also experience significant pain, and that in turn can lead to depression. So it's a very disabling disease in itself. And around 1 to 2% of the general population, that's 2 to 3 million adults in the US, and about 500,000 adults in the UK have psoriatic arthritis. So we've been trying to develop protein biomarker tests that can support the diagnosis of psoriatic arthritis. And the way that we've done that is to do discovery, where we've taken existing panels that we've had, candidates from the literature, and done our own discovery experiments to come up with a panel of candidates that we then develop MRM tests for and analytically validate those tests. And over a number of years, we've been doing that in psoriatic arthritis and a number of other arthropathies, including rheumatoid arthritis and juvenile idiopathic arthritis, and come up with a panel of around 206 candidate inflammatory arthritis biomarker proteins that we target by measuring 432 peptides. And we do that in an assay that we call the paprika method, and this just shows some of the properties of that assay. The important thing is that the mean peak area CV, so that's the ability to quantify the peptides, is less than 10% for all 432 peptides. So if any of these peptides prove to be valuable biomarkers, the assay has been developed sufficiently for them to become incorporated into a test. So that's what we've done. We've taken that panel and we've tried to clinically validate it in cohorts of patients. I'm just going to show a snapshot of one of those, and that's a cohort of patients that have psoriatic arthritis versus rheumatoid arthritis. And in a modest number of patients, so just under 200 in total, we were able to measure the peptides in the paprika panel and show that a subset of them can discriminate between psoriatic arthritis and rheumatoid arthritis with an area under the curve of about 0.9. So that's a very high area under the curve, a 90% probability that we can discriminate between those two diseases. And so we've spent some time now surveying rheumatologists as to whether that would be something that would be valuable to incorporate into their clinical pathway. And it seems that they universally agree that it would be very valuable because of the challenges associated with diagnosing psoriatic arthritis, and that it would increase the time, it would decrease the time it takes to make that diagnosis and allow them to intervene at an earlier stage in the disease. So the next steps, because this still is at an early stage, is for us to use Oliver's connections in the GRAPPA network to access high-quality clinical samples and their clinical expertise and undertake a clinical validation study. So we're in the process now of designing and getting access to the samples for that clinical validation study. And then we very much hope that if that clinical validation study proves the power of the test in being able to discriminate psoriatic arthritis from rheumatoid arthritis, it will be adopted and disseminated by the KOLs. And of course, we're really fortunate to be connected and inculcated with this GRAPPA network, because they're the group of clinicians who set the clinical guidelines for psoriatic arthritis. So a sort of a whistle-stop tour of some of the work that we've been trying to do and some of the limitations that may exist. So I'd just like to end now with an overview, with how we might deliver these protein biomarkers for chronic diseases. And it turns out, in collaboration with the Gemelli Hospital in Rome, we've identified that in our initial test delivery model, where the sample is sent to this central Atturos laboratory for performing these assays, getting the sample to the laboratory is the most challenging and expensive part of the process. So with the Gemelli and with colleagues in Neoterics, we've been exploring whether serum placed on quantitative sampling devices called Mitra tips could be used as a way of shipping the samples at ambient temperature, dry and at ambient temperature, to the central laboratory. And in early experiments, we were able to show that we can perform these tests off serum that's been dried on the Mitra tips. And on the right-hand side, you can see there that the peptides that we measure are stable at ambient temperature on the Mitra tips for a significant amount of time. So we think this provides an important logistic step in terms of getting samples to a laboratory that can process and undertake these diagnostic tests. So this is just a summary of where Atturos is up to in terms of its pipeline of tests in psoriatic arthritis, in prostate cancer, and through a test that it's licensing in from a company called Proteomics International in Australia, the test Biomarker D in kidney disease. And it shows how we're trying to progress these through this pipeline. So I'm just going to end with some key messages. I think it's clear to all that many new tests are needed to support this era of precision medicine, to go from the very ineffective approach that we have at the moment to perhaps the vision that Leroy Hood anticipates. And the expectation is that they will measure multiple biomarkers, not single. And I suppose that they will also measure proteins as well as genes and genomics and metabolomics. So we think protein tests are good, but when combined with other omics measurements, whether that's genomics or metabolomics or lipidomics, they will together add value and be better. It's clear that mass spectrometry has made a huge impact on many clinical tests, and I haven't had a chance to describe those small molecule tests where it's routinely used, but we believe it's poised to do the same for new multiplex protein tests. The other message is, well, we know it's challenging, but we believe it's achievable. And indeed, I think we're possibly not alone in believing that it's achievable, but it still is proving extremely challenging. So I think it just remains for me to acknowledge my colleagues who've made all of the psoriatic arthritis work in particular possible, and specifically Oliver Fitzgerald, and the team of clinical, scientific, statistical, and external collaborators that we're working with, but also to thank Agilent Technologies for providing us and working with us to develop this biomarker workflow and the platform on which we perform these measurements. Of course, the most important individuals to thank are the patients, because it's their samples that we use in this development process. And we're really fortunate that the GRAPPA network includes significant representation from the patients. And we engage with them at the very earliest stages and keep them engaged with everything that we do. So finally, I'd just like to thank the early colleagues in Atturos, Agilent, PerkinElmer, Neoterics, our colleagues in the Gemelli Hospital and Merck, who've helped us develop some of the analytical validation and the steps required for analytical validation. And then just show you the scale of Atturos as it stands at the moment. It's still very embryonic, as you'll see. So this is the small Atturos team who are really focused on developing this next generation of protein biomarker tests. So I'll just leave it to thank you then for listening and I hope then to be able to answer some questions or any questions that you might have. Thank you.
Hello, my name is Cliff Dacso, and I'm a professor of molecular and cellular biology and medicine at Baylor College of Medicine. And with my colleague, Dr. Arun Sreekumar, we're going to talk to you today about metabolic reprogramming in cancer and specifically focusing on biomarker development. I'm going to spend the first couple of minutes talking in general about screening for malignant disease as well as for benign disease. and the criteria that we use for screening and for the development of a test using metabolomic criteria. So the first criterion for a screening test is it has to be a significant disease. There are a lot of tests for which good screening tests would be available, but the cost and effort required to perform those screenings make it not viable. So the second part of that is that the disease has to have a high prevalence. Why is that? Well, one of the reasons for that is Bayes' theorem, which says that the posterior probability, that is the likelihood of a test giving you useful information, has a strong dependence on the pretest probability. So you'd like to have a high enough prevalence of the disease in the community to circumvent the abyss of Bayes' theorem. The third thing is that early treatment would be beneficial. That is to say, in illnesses where the disease itself is not amenable to treatment, or treatment does not make much of a difference, why do the screening to begin with? We have to ask for a test that has a high degree of specificity and sensitivity, and although not on this slide, by implication has to have a reasonable positive test predictability. And then the last thing is the screening has to have a low morbidity and low cost with follow-up testing available. So these are roughly 6 criteria that are generally used to decide for whether a screening test should be inaugurated or not. The converse of that is a list of 4 items of when screening is not favored. If the disease is rare, as I said before, it's not serious, or there is no effective treatment or public health import, then one would not embark on a screening program. If the population prevalence is unknown, one can still do screening, but we have to use the right mathematics for understanding the ability of a test to give a diagnosis, or the converse, a negative to be a true negative. If there is no benefit to treatment, that is, no intervention is indicated, then there's really no reason to do screening either. And lastly, and this probably is an absolute, specificity and sensitivity cannot be determined. In that case, it really wouldn't pass muster as a valuable screening test. From the point of view of prostate cancer, you would think after all these years we would have this one locked in, that we would know exactly how to do screening for prostate cancer, when to do it, and who to do it on. But at this point, other than limiting prostate cancer screening to men, there's a lot of dispute still in the literature. The U.S. Preventive Services Task Force has strong recommendations regarding prostate cancer. And these recommendations are, I should say at this point, relatively fluid because actual practice varies. For the most part, after age 60, men are asking for screening tests for prostate cancer. I certainly experienced this in my clinical practice, that I might be, even in the setting of shared decision-making, the people are requesting prostate cancer screening. They'd like to know why. Their expectations are that we'll be able to catch the disease early and intervene. Another controversy is the heterogeneity of prostate cancer. We are all familiar with the Gleason categories and their implications for clinical practice, but there's more to it than that. And the heterogeneity of prostate cancer and the impact of genetics is still under study and somewhat controversial. Number 4 is perhaps the most important, which is there are disparities of access and treatment. Different populations have different access to urologic care for biopsy and cancer care for treatment. These are geographically distinct, they're economically distinct, and these disparities have a major impact on whether or not to embark on a cancer screening effort. And then lastly, there is still controversy about treatment strategy. Is radical prostatectomy the treatment of choice? Is external beam radiation the treatment of choice? How about watchful waiting or expectant observations, where we do no active intervention, but the man needs to come back for repeated cancer diagnosis? All these are controversial. There are large studies pending, and we ought to have information on this in relatively soon, but we don't have it right now. One of the most important aspects from our perspective, from our research program, but also from public health and clinical aspects, is that African American men have prostate cancer earlier and worse than European American men, or Asian American men for that matter. And this chart from 2017 shows that beginning at the moment of diagnosis and continuing on as far as the eye can see, the prostate cancer specific mortality is higher in black men than it is in all non-black men. And this graph informs our target population, where, thinking about those screening criteria that I talked about earlier, the money really is. We have a population where intervention would be important, where we have a test with a known positive predictive value, and we have a population of high prevalence. And if you throw in other aspects like family history of prostate cancer, this curve gets even more acute and screening is even more indicated. So what are the opportunities for the metabolic characterization of these cancer diversities? Because in African American men, just like in European and Asian men and non-Hispanic whites, the metabolic characterization of cancer diversity determines the treatment. And in this situation, an appropriate screening test will give this type of information to inform further treatment, whether that be a chemotherapy strategy, an immunotherapy strategy, radiation, where we can predict radiation responsivity. We'd like to be able to make some predictive statements about response to therapy, and even more importantly, because we know the biology of this disease, it's important to be able to have a test available that will give us a high degree of ability to perform surveillance for recurrences. So in terms of the vulnerable populations for prostate cancer, it is a large number of people. A large number of these people will require primary tests, and even more will require confirmatory tests, and even more will require primary tests. And we see in the future that there's going to be a significant growth in this population. What are going to be the drivers of that growth of utilization of the screening test? One is aging of the population. Prostate cancer is not a disease of youth. As time goes on, there are going to be more and more effective treatments. There are a number of treatments now in clinical trials. More are coming. The pipeline is large. It's going to be really critical, as I talked about earlier, to be aware of differential populations, not only in terms of their susceptibility to disease, but also their education about the disease. Because for an effective screening program to be embarked upon, we have got to have a high degree of awareness in the population that screening would be beneficial for them. And then lastly, the effectiveness of testing is a critical issue, one that really drives the entire program. So now I'm going to turn this program over to my colleague, Dr. Arun Sreekumar, professor in the Department of Molecular and Cellular Biology at Baylor, who will talk more about the metabolomics.
Thank you, Cliff. That was a great introduction on prostate cancer and the clinical management of the disease. I'm going to be now talking about how we have used metabolomics as one of the platforms to try and understand some of the biological changes that are associated with prostate cancer in African American men, and how we plan to use this information to develop markers for early detection of the disease. So to begin with, as you all know, metabolomics is basically is a technology that's used to detect the steady state levels of small molecules and metabolites. So in our study, we have used a mass spectrometry based approach to look at metabolic alterations. And our first step in the study was to look at metabolic alterations that were associated with prostate cancer of African American men, which here is abbreviated as PCA, relative to the metabolic profiles in the benign adjacent tissue. So the benign adjacent tissue is basically the unaffected, pathologically unaffected part of the prostate that is lying right adjacent to the tumor. So what I'm showing you here is a snapshot of all the metabolic alterations that were identified. This is a heat map wherein shades of yellow represent metabolites that are elevated, while shades of blue represent metabolites that are downregulated. And metabolites are arranged on the y-axis or on the rows, and the columns represent the samples. So a quick look at the heat map tells us that there are metabolic alterations that are associated with prostate cancer in African American men compared to matched benign adjacent tissue. And if you look a little further, and I'm specifically just highlighting names of certain metabolites that belong to a particular pathway, which in this case happens to be the Methionine-Homocysteine Pathway, also known as the one-carbon metabolism pathway. And you can see that majority of the metabolites in this Methionine-Homocysteine pathway seem to be elevated in prostate cancers in African American men relative to the adjacent benign tissue. So one way to look at this data is to represent it in a pathway context, which is what I have done in the next slide, which is basically a network diagram that shows you how these metabolites fall into various pathways. The major pathways that are most highly enriched are shown in red circles with a blue background. And the nodes, the bridges, they basically represent the interaction between the various pathways. Nevertheless, the take-home message here, if you look at it very carefully, is that among the many pathways that are enriched, one of the ones that was most highly enriched was the cysteine-methionine metabolism pathway, which I'm showing you in the red fonts right in the center of this picture. So going to what's cysteine-methionine-homocysteine pathway? Methionine-homocysteine pathway is represented in this particular slide, where methionine that's derived from the diet is converted to S-adenosine methionine, which all of you know is a methylation currency. And when S-adenosine methionine donates its methyl group, it gets converted into S-adenosine homocysteine, that then stoichiometrically breaks down into homocysteine and adenosine. So based on the heat map that I showed you a couple of slides prior to this one, I had shown you that some of the metabolites in this particular pathway were elevated, and I had highlighted metabolites, namely methionine, homocysteine, adenosine, cystathionine, and so on. So the take-home message here from the first set of profiling experiments that we did was that there was elevated levels of methionine and homocysteine in the tissue specimens that were obtained from which are either from prostate tumors and related to the adjacent benign tissue. So we went ahead and then tried to look a little deeper into this. So in the next slide, we did a validation of these findings in plasma. And here I'm showing you three box plots, and these represent two different patient cohorts. On the left, I'm showing you the box plot for the levels of homocysteine, the relative levels of homocysteine in a case control cohort. And these involves African-American prostate cancer patients and African-American case controls. And you can clearly see here that the levels of homocysteine are significantly elevated in the plasma of African-American prostate cancer patients relative to the case controls. Panel B is an independent cohort, which is called what we call as a biopsy cohort. And here, this is derived from patients who come into the clinic with elevated levels of PSA and go through biopsy to detect the presence of cancer. So the samples here are collected from a clinically challenging population of patients who have elevated levels of PSA and who have abnormalities that require them to go through the biopsy. And the samples are collected prior to biopsy. And the results are then compared to the biopsy results. And as you can see here, we have a biopsy positive cohort and a biopsy negative cohort. All patients in this cohort are African-American men. And it's clear that individuals who have a positive biopsy have higher homocysteine levels compared to individuals who have negative biopsy within this cohort containing African-American men. Panel C is, again, the case control cohort that I described in panel A, but comparing the levels of homocysteine in African-American prostate cancer, in African-American men with prostate cancer compared to European-American men with prostate cancer. Here again, you can see that African-American men with prostate cancer have significantly elevated levels of homocysteine compared to their European-American counterparts. This kind of, again, verified our original findings that homocysteine levels seem to be elevated in African-American prostate cancer, both at the level of the tissue that I showed you earlier and at the level of plasma. So the next question we had was, is there an association between the elevated levels of homocysteine and the West African ancestry? That's what I'm showing you in the next slide here, where you can clearly see that when you take all samples into consideration, both and this analysis was done in the case control cohort. When you take both the cases and the controls into consideration, there is a significant correlation between West Africa ancestry and elevated homocysteine levels, as I'm showing you in panel A. And in panel B, this association remains when you only look at the prostate cancer cases, but is not evident when you look at the controls, showing that there is a correlation between West African ancestry and homocysteine levels in the serum of the patients who have prostate cancer. The next interesting finding is when you further dig into this a little deeper, you'll see that the West African ancestry has an odds ratio of 18.5 to have high levels of plasma levels of homocysteine. That means West African ancestry per se increases the probability of having elevated plasma levels of homocysteine in these individuals. The next slide shows you a little bit about some of the reasons that would explain this elevated levels of homocysteine in these patients. Here in panel B and panel C, I'm showing you data from tissue microarrays, where we are comparing the expression of two proteins. In panel B, I'm showing you the expression of betaine hydroxymethyltransferase, which is an enzyme that remethylates homocysteine back to methionine. And on panel C, I'm showing you the expression of cystathionine beta synthase, which is an enzyme that takes homocysteine and converts it to cystathionine. In both cases, as you can see, the expression of these proteins in African-American prostate cancer is significantly lower compared to European-American prostate cancer. This could partly explain the elevated levels of homocysteine that we find in African-American prostate cancer patients that I've shown you in the previous slides. Having identified this, the goal of our research is to see how we can use this information to develop markers that can be used in a non-invasive setting to identify or to detect the presence of cancer or the presence of prostate cancer in African-American men. So for this, we have a collaboration with Agilent. There is a ongoing collaboration with Agilent and Baylor College of Medicine, the goal of this collaboration is to develop a high-throughput, non-invasive LC-MSMS-based metabolomic signature for early detection of prostate cancer in African-American men. And as Dr. Dacso mentioned earlier during his talk, African-American men with prostate cancer have 2 times higher mortality rate compared to European-American men. And the patients that we are using in this biomarker development strategy, all these patients have a significantly elevated levels of PSA, which is clinically significant, which in this case I've indicated as greater than 4 nanograms per ml. and or have an abnormal digital rectal exam, either of which necessitates that they go through a biopsy for confirmation of presence of cancer. In the case for this study, we have collected all the samples prior to biopsy. An important thing that we all need to note here is that thus far, there are no biomarkers defined for early detection of prostate cancer in African American men. And as Dr. Dacso indicated, early detection of prostate cancer in African American men could be beneficial because it will give a window of therapeutic intervention that would benefit these patients wherein the tumors are pretty aggressive. So the question we have is, can plasma levels of these metabolites distinguish biopsy-verified prostate cancers from biopsy-negative controls? To do this, we have done two sets of pilot studies, both involving plasma samples collected at the time prior to biopsy, as I mentioned in my previous slide. The first pilot set contains about 47 samples, of which 20 are from African-American biopsy-positive patients, 17 are from African-American biopsy-negative individuals, and 5 each are from European-American biopsy-positive and biopsy-negative individuals. In pilot set 2, we have an additional 72 samples, which include 34 from African-American biopsy-positive cases, 24 from African-American biopsy-negative individuals, and 7 each from biopsy-positive and biopsy-negative European American individuals. Again, this is a pilot study, but it basically lays the foundation to show that metabolic markers would have clinical value as diagnostic markers. So next slide shows the preliminary data that we have obtained using a multi-metabolite panel. This includes multiple metabolites that we have identified in our study. And as you can see, this is compared with PSA on the right for the population of patients I just described in my previous slide. And it's evident that the metabolic panel per se has a superior area under the curve with an ROC of 0.71 for early detection of biopsy-positive status in African-American men compared to PSA, which has an ROC of 0.56, which is just equivalent to about the toss of a coin. So this pilot study that we have begun in this collaboration, basically, we hope to carry this forward, and we hope to develop a biomarker panel that can be clinically utilized that would benefit African-American men and help early detection of the prostate cancer in these individuals. The team that's involved in this investigation involves Dr. Bert O'Malley and Dr. Cliff Dacso, myself, Dr. Nagireddy Putluri, and Dr. G. Godhe. Thank you.
Well, thank you all for your informative presentations. We really appreciate it. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll get to your question. So let's get started here. The first question is going to be for Dr. Steve Pennington. About how long do the protein biomarker MRM methods take to run?
Lovely. So thank you for that question. It's a good one. It obviously depends on the number of proteins that we're measuring. Peptides, the shorter the assay is itself. Typically in the early, we try to keep the assays include as many proteins as possible. But as we're moving towards refining the panels, the assays that people can show. That's a long answer to a short question, and I haven't answered it yet. I would to say that the assays take anything between 5 minutes to minutes to run spectrometer.
Thank you. And another question following up is, how is psoriatic arthritis currently diagnosed, and where will these tests be performed? In hospitals?
Okay, again, thank you for that question. At present, present, psoriatic arthritis, there are no diagnostic tests for it, so it's a clinical assessment that's made, and predominantly the assessor and the diagnosis depends on the skills of the assessor and their experience, and they are looking for signs of inflammatory musculoskeletal disease in the presence of involvement and skin psoriasis. But those can be very hidden features, and indeed psoriasis may not be present at the time of presentation. And so it is a very challenging diagnosis to make, and that's why we and rheumatologists have been working with believe that a test that could aid in the diagnosis would be a very valuable addition to their skill set. Sorry, the other part of the question was?
Where will these tests be performed?
Yeah, so again, that's interesting. I think that our expectation is that in the first instance, this will be performed in central laboratory facilities, but ultimately they may be out into hospital laboratories if the instrumentation, the mass spectrometry-based instrumentation, can be provided in a format that's amenable to hospital laboratories. And of course, there are mass spectrometers currently being used in hospital labs for measuring small molecules like immunosuppressants and things like vitamin D. And I suppose we anticipate that in the future, those mass spectrometers or equivalent mass spectrometers in hospital labs would be able to analyze the panels of proteins that we're developing here.
Thank you. Our next question is going to be for Arun. Arun, what methods were used to come up with these pathways and visualize them?
Yeah, that's a great question. We used a methodology called NetGSA. It's a network-based, network topology-based method that basically takes into account the underlying interaction between the biomolecules in a network setting. And this particular topology enrichment method basically uses a mixed effect linear regression framework and basically conducts a formal hypothesis test to come up with pathway enrichment. So this is basically called NetGSA. And we basically visualize this using the R program. And we basically used a program within the R console, which is called Kamada Kawaii.
And is there a signature for castration-resistant prostate cancer?
Yeah, great question. So what I spoke earlier during my, what I presented earlier was basically trying to look at prostate cancer that was organ confined, which is what we call as a localized prostate cancer. And castrate-resistant prostate cancer is an advanced disease, which is basically associated with, as you know, resistance to second-generation anti-androgens. So when the prostate cancer recurs or comes back, which is what we call as biochemical recurrence, and at that point, you know, when the patients start getting treated with second-generation androgen deprivation therapy, and when they become resistant to that, that's when we call castrate-resistant prostate cancer, which is in fact considered a lethal disease. And we have done analyses to look at metabolic profiles in castrate-resistant disease compared to, for example, organ-confined disease. And we see significant alterations or signatures that are important signatures that are specific to castrate-resistant disease. Again, it all depends on the drivers that are associated with CRPC or castrate-resistant disease. For example, we do see in case of the CRPC, we do see changes in lipid metabolism to be happening significantly. We also see changes in carbon utilization, where, you know, and there's a switch from being an amino acid dependent tumor, which is what we call as an organ confined tumor, to a sugar dependent tumor, which is what we see in case of CRPC.
And one more, do you think that there is a test to predict recurrence?
At this point, there's not really a good molecular test out there to look at the predict biochemical recurrence. Most of these predictions are using clinical nomograms. There are efforts that are being put in this field, both using genomics, proteomics, and metabolomics to look at markers that could be, or molecular signatures that could be used to predict the probability or the potential of a given patient having biochemical recurrence, but that's still in early research stage.
Well, thank you. Thank you. Our next question is going to be for Arun. Can metabolomics be applied to following men with prostate cancer?
Yes, I think there is a huge potential for metabolomics to be used in the biomarker world, especially for following the progression or the trajectory of the disease. The important thing about metabolomics is it basically reflects a physiological state of an individual at any given time point. And so if it's easy, it's non-invasive, it can be detected in biofluids. And so what that allows us to do is it allows us to do longitudinal testing and allows us to develop signatures for progressive disease versus disease that is indolent. And so I think definitely this is still a field that's really coming up, and I think there's a lot of potential in using metabolomics trying to monitor disease progression.
And do you anticipate that metabolomics will allow early detection of prostate cancer in African American men?
Well, I guess the entire hypothesis there is that there is a biological difference that exists between, African American men and prostate cancers of African American men and European American men. Although there are other factors that also could contribute to this disparity, which include socioeconomic status and so forth, health access to healthcare and other things. But we do see biological differences, which in this case is differences in metabolic profiles. And we do see some alterations in pathways, which we believe could be exploited to develop metabolic tests for early detection, and that's the attempt.
Not hearing. Okay, well, unfortunately we won't be able to get to Cliff, but gentlemen, again, I want to thank you so much for your time and your important research. We really appreciate you. Before we go, I'd also like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed to the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand through the end of June of 2021. Until next time, goodbye.
Thank you. Thanks. Bye.